day of a prestigious GAN course on Advances in Experimental Therapeutics, and we are thrilled to begin this conclusive day with the same energy and academic zeal we have maintained throughout the week. This course has been a significant platform for knowledge exchange, research insights, and international collaboration. On behalf of the entire organizing team, I extend my heartfelt thanks to one of you, our esteemed speakers, coordinators, and participants for making this event a resounding success. Your efforts, vision, and guidance have shaped this course a truly impactful learning experience. To begin today's academic proceedings, we invite our distinguished international expert, Professor Sanjay V. Malhotra, to deliver the lecture on the case studies.
No, I don't watch.
No, you have to click on duplicate.
I will, यहाँ पे ना, I'll go there now. Thank you. Thank you, Tanya. Good morning, everyone. So what I thought probably I will share with you is now I showed you a few examples of, as I was telling, experimental therapeutics. Hopefully, with what I have shared with you, you realize the fact that it's a multidisciplinary enterprise. It's a multidisciplinary field where many disciplines, many things come together to develop a therapeutics, even to understand how the therapeutics works. So what I thought now in the final, this lecture is to bring all of these things together to show a case study where you will see the applications of many of these utilities, many of the modalities, applications that I have been telling individually, from imaging to therapeutics to analysis to PK and all of that, how these are actually employed, used for developing new therapeutics. So my lab, my work, as I showed you, as I told you, I'm a trained chemist, synthetic organic chemist. I did my bachelor's in chemistry, pure chemistry, and then applied chemistry, and then again synthetic chemistry, catalytic chemistry. So my training has been purely on chemistry. But as I mentioned, as a chemist, I had the curiosity, and fortunately, the opportunities to be picking up those projects which taught me a lot. Because not that you enter into project and you सब कुछ आपको आता ही है, सर्व संपूर्ण कोई नहीं है. So I started as a chemist. The reason I'm sharing with you is how this journey began and what we do. So now in my lab, for example, what we focus on is in the area which is in the black. So top bar पे जो आप देख रहे हैं, it is the process, the path जो drug discovery, drug development. So यहाँ से target identification is something that, for example, for a disease you have a target. Target could be initially what disease type is. Let's say you're interested in a type of cancer or neurodegeneration. In there, then you want to understand what the biological target is. So biologists working with you will say, okay, हमने जो study की है, this seems to be the target. Sometimes they do, sometimes they don't. Presume based on the data that they have. Once they do that, my lab also does that, but once they do that, after that it becomes whole experimental therapeutics part. To identify which modality will work, how it will work, small molecules will work. So I have written here small molecule, that's not the only thing we do. We do peptides, antibody drug conjugates, mostly these modalities that we work on. So this path that shows is you are do the high. Once you have identified what your target is, you develop an assay. As I was telling you कि assay होना चाहिए कि identify करने के लिए कि protein को आपने follow करना है. You develop an assay, and then you screen compounds, whether they are known compounds or unknown compounds. You screen thousands of compounds, you design compounds, and I'm going to share with you how we designed the compound, from where we started based on the knowledge of that. And then you again lead optimization. एक बार आपने हिट निकल आया, उसको ऑप्टिमाइज करना लीड, कि अब उसको जो है ना आपको यहाँ, रिमेंबर आई शोड यू एरो टारगेट में दूर है उसको लेके आपने बुल्स आई एकदम सेंटर में लेके आना है टारगेट पे. दैट इज़ द ऑप्टिमाइजेशन प्रोसेस. देन लीड प्रोफाइलिंग, सारा एडमी डॉक्स. अब उसका प्रोफाइल आपने चेंज करना है कि इट इज़ सेफ एंड एफिकेशियस and something that is really going to be helpful. Due to the lead profiling, then it enters the clinical part, जो कि मैं yesterday, as I told you. So my lab, our center works on all of these things, and we employ many different techniques. So we design molecules, we use chemical biology to understand the biology, the tools that we create. These tools are the molecules that you create, generate, use to study biology are called chemical tools or probes. They are probing the biology. We use genetics. Genetic level पे उसकी क्या information है to study that. We design de novo scaffolds, new compounds. We take motivation from what we learn from nature या जो published में है, but then we see if that is helpful. Otherwise, we design as a synthetic chemist, medicinal chemist. We design our own modification करके कि how it will help us to understand the biology of the disease. We do a lot of imaging, and as I told you that we have a whole cyclotron and we have a whole radio pharmaceutical synthesis enterprise. We do a lot of imaging. We design molecules so that you can image the in vivo studies and make sense of that. We study radiation because radiation, when you radiate, it's a DNA damage when you do this. So when you do the DNA damage of a target in a cell, whatever, let's say cancer cell hai, uske andar if you're doing, what happened? How do you manipulate the radiation in combination with the drug? So we do a lot of those studies. And then in this process, if we identify a hit compound or a compound which is really helpful, then we jump on this path. जो नीचे है सारा, this is what the pharma industry does. कि आपने एक ड्रग लिया, जो लीड को डिस्कवर किया, you discover, and then you optimize the lead and you keep on iterative process जब तक कि जैसे as I was telling you, उसकी properties क्या हैं, admitox, solubility, bioavailability, non-toxic safety, you keep on studying these things and optimizing. उसको आप पॉलिश करते रहते हैं, उसको एडिट करते रहते हैं, उसके एनालॉग्स बनाते रहते हैं. सो वी डू ऑल ऑफ दीज थिंग्स. और एक बार सक्सेसफुल हो गया, then it enters the drug development stage. So we do most of this in-house in our center, in our institute, in my lab. So when we do this, we employ many of these techniques. As you see here, we pharmacology, genomics, imaging, and immunology. We study discovery से लेके preclinicals, IND enabling से वहाँ थे. We use bioinformatics, biostats, biomarker development, and of course we are educational. We are an educational institute, so education is part of whatever we do. And as you see here, before it enters clinical phase, right from the beginning we are interacting with all these experts area. We do a lot of this in my lab, but as and when needed we engage. So it's a collaborative enterprise. No one person can easily may have वो तो उससे अच्छा तो हाथ रखे के तथास्तु और हो जाएगा काम. इसमें काम करना पड़ता है. So this is a multidisciplinary enterprise that we do together. So what I'm going to share with you now an example of how in my lab our group started employing many of these techniques and different pieces जो मैं अब तक आपको 4 दिन से बताता आ रहा हूँ, हमने सबको कैसे मिला के एक लीड कैंडिडेट को कैसे बनाया. So you'll see a single candidate निकालने के लिए किस तरह से सारी जितनी भी जो एक्सपर्ट जो डिसिप्लिन्स हैं, नॉलेज हैं, it comes together to do that. Now what is cancer? So we know that cells divide, and when they divide they accumulate DNA damage. DNA का डैमेज होता है क्योंकि ब्रेक होता है, डिवाइड होता है, रिपेयर होता है, एंड मोस्ट ऑफ द डैमेज इज रिपेयर्ड. कुदरत ने बनाया इस तरह से है कि द डैमेज गेट्स रिपेयर्ड. बट इफ इट इज नॉट रिपेयर्ड, सो बेसिकली क्या है, यू नो द केमिकल रिएक्शन. I thought there was a knock on the door. So if it is not repaired, it's a basically chemical reaction, bond breaking, bond making. So there is a kinetics involved. आप अगर physical chemistry अगर आप याद करें तो उसका जो है there is a rate of reaction. Bond break होता है, bond बनता है, bond break होता है, bond बनता है. It happens in picosecond speed से. And if for whatever reason, so and most of the time these things are repaired. Occasionally there is a bond जहाँ से, as I was telling you, DNA what is many base pair, many base pair, amino acid, nucleic acid, carbohydrate, fatty acid. I showed you polypolymer. वो base pair हैं जो connect करके प्रोटीन्स बनाते हैं, आरएनए बनाते हैं, डीएनए बनाते हैं. सो कुदरत की रचना ऐसी है कि इट हैपन्स एग्जैक्टली एट द सेम प्लेस अगेन अगेन, राइट. सेल्स इसी से क्रिएट होते हैं. अगर नहीं हुए, इट अटैच समवेयर एल्स, इट बिकम्स, सो जहाँ से बॉन्ड बनना चाहिए अगर वहीं जाके नहीं लगा, 1 in 10,000 टाइम्स. जस्ट इमेजिन, अराउंड 1 in 10,000 टाइम्स. आप यहाँ बैठे हैं, हम सब यहाँ हैं इस रूम में, हर एक के सेल्स के अंदर वो डीएनए जो है ब्रेकिंग मेकिंग चल रहा है, सेल फॉर्मेशन इज गोइंग ऑन. सो यूजुअली जो एज पर द स्टडी शो के अराउंड 1 in 10,000 टाइम्स वो बॉन्ड सीधा वही नहीं लगता है. इट्स अ केमिकल रिएक्शन. आपने देखा अमीनो एसिड का स्ट्रक्चर क्या है, इट फॉर्म समवेयर एल्स. एक्सक्यूज मी. व्हेन दैट हैपेंस, वो अब उसने थोड़ा स्ट्रक्चर चेंज हो गया, इट्स कॉल्ड म्यूटेशन. वो म्यूटेट कर गया, ठीक है. सो दीज mutations can occur anywhere on the DNA, right? DNA इतना बड़ा है, सारे functional groups लगे हुए हैं, amino acids वगैरह, so that mostly and जो mutations इकट्ठे होते हैं, these mutations are called passenger mutations. वो साथ में चलते रहते हैं, आपका सेल डिवीजन हो रहा है, वो साथ में चलते जा रहे हैं, there isn't much really happening, they are harmless. आपके सेल के अंदर वो साथ में चलते जा रहे हैं. Occasionally what happens is these mutations become important and really of interest to the cell. सेल की ग्रोथ के लिए वो इम्पॉर्टेंट होता है, सेल फास्ट ग्रो करने लगता है. नाउ व्हेन इट स्टार्ट्स ग्रोइंग फास्ट, इट स्टार्ट्स टू दोज म्यूटेशंस, धीरे धीरे वो कोडॉन्स जो है इकट्ठे होने शुरू हो जाते हैं. एंड स्लोली दैट ग्रोथ becomes faster. नब कुदरत ने बनाया है आपका इम्यून सिस्टम, आपका फाइटिंग मैकेनिज्म जो है इंसान की बॉडी में, बायो क्रिएशन बॉडी में, वो इम्यून सिस्टम आइडेंटिफाइज एंड ट्राइज टू क्लियर इट अवे. वो एक्सक्रीट कर जाता है, बाहर निकल जाता है, क्योंकि इम्यून सिस्टम रिकॉग्नाइज दिस इज समथिंग फॉरेन एंटिटी, वो उसको बाहर कर देता है. बट समटाइम्स दे आर सो रिजिड, क्यों बाहर नहीं होते, दे कीप ऑन collecting, and this becomes a lesion. And but sometimes these lesion again evades the immune system को, immune system को वो धोखा देके आगे निकल जाते हैं and eventually grow, and it's called cancer. क्योंकि आपको पता नहीं है कि म्यूटेशन क्या हो गया, कहाँ से हो गया. द सेल इज ग्रोइंग फास्टर, इट्स नॉट द नॉर्मल सेल, नाउ इट्स ग्रोइंग फास्टर. इम्यून सिस्टम को बीट करके वो आगे निकल गए. सो दैट इज ग्रोन. नाउ व्हाट हैपेंस इज आइडेंटिफाइड, पेशेंट गोज टू द डॉक्टर, डॉक्टर लुक्स एट, आइडेंटिफाइज कि किसी एक जगह पे उन्होंने लीजन उन्होंने नोट किया, डायग्नोज किया. डॉक्टर के टूल बॉक्स में जो भी दवाइयाँ हैं, जो भी अप्रोचेस हैं, वेदर सर्जिकल अप्रोच है, रेडिएशन अप्रोच है, कीमोथेरेपी है, इम्यून थेरेपी है, जो भी अप्रोचेस हैं उनका कॉम्बिनेशन है, देने की कोशिश करते हैं. एंड देन व्हाट हैपेंस इमीडिएटली, उस समय जो अटैक होता है कि दोज कैंसर सेल्स डाई, निकल जाते हैं, और जो डायग्नोस होता है उसमें नज़र नहीं आते. बट उसमें कुछ टाइम बाद में फिर से पेशेंट जाता है. नाउ डॉक्टर सीज कि इट्स नॉट एट द सेम प्लेस, बट नाउ इट हैज स्प्रेड एट अदर पार्ट्स ऑफ द बॉडी, बिकॉज यू कैन नॉट डायग्नोज. बट इफ द सेल थोड़ा सा भी रह गया है, they grow fast. वो grow करके, and then doctor will give another whatever is available in his or her kitty toolbox. Again it suppress किया, and after some time initially it's suppressed and then it comes back. You try again, doctor tries again, eventually after some time it becomes the race, क्योंकि जितना डॉक्टर के उसमें जो तरीके थे, सर्जिकल था, रेडिएशन था, कीमो था, इम्यून था, जो गया वो कॉम्बिनेशन सब कुछ ट्राई कर चुका, बट इट्स स्टिल इवेट्स, एंड दैट्स व्हेन यू हियर द टर्म कि दवाई काम नहीं कर रही, राइट? ये आई एम ट्राइंग टू टेल यू एज ए साइंटिस्ट, समझने की कोशिश कीजिए कि दवाई काम नहीं करी उसका मतलब क्या है। मीन्स दिस कि जो दवाइयाँ दी थी, जिस चीज़ के लिए दी थी, राइट, उसके बावजूद वो इवेट करके ग्रो करा है। सो एंड व्हाट से कि पेशेंट हैज़ डेवलप रेजिस्टेंस टू द ट्रीटमेंट, राइट? दीज आर द टर्मिनोलॉजी, कॉमन टर्म यूज। साइंटिफिकली क्या हो रहा है, जो ड्रग्स बनाए गए थे, रिमेंबर नाउ यू कनेक्ट दिस टू इन पास्ट 2-3 days व्हाट आई हैव बीन सेइंग, ड्रग अगर बनाया है उस टारगेट के लिए नहीं बनाया है तो शुरुआत में तो इतना कीमो इतना स्ट्रांग होता देंगे तो कुछ दिन के लिए सेल वाले को मार देगा क्योंकि इट्स वेरी है, बट उस टारगेट को नहीं दबाया है तो कहीं से भी निकल कर नजर नहीं आता, फिर से निकलना शुरू हो जाएगा। सो देयरफॉर डिजाइनिंग अ ड्रग विच इज स्पेसिफिकली विद दैट प्रोटीन, नाउ यू सी कि हाउ इट्स इंपॉर्टेंट। अदरवाइज वो इनिशियली तो असर होगा क्योंकि अब इतना टॉक्सिक चीज़ दे रहे हैं, दीज आर हाईली टॉक्सिक केमिकल्स, and they initially they are bombarding and there are side effects that you see, unfortunately. The patient goes through a lot, body loss, weight loss, hair loss, all that, क्योंकि वो टॉक्सिसिटी है ना, उसका साइड इफेक्ट है। बट उसके बावजूद भी जो ड्रग दिया गया है, जिस टारगेट जो है उसके लिए अगर नहीं बना है तो पेशेंट गोज थ्रू ऑल दिस ट्रबल, ऑल चीज़ के लिए कुछ टाइम के लिए सप्रेस हो जाता है, बट टारगेट के लिए तो बना नहीं है ना, टारगेट उसके लिए वो अटैक हो नहीं रहा है, सो इट कम्स बैक and we call this resistance. Now over the years have been made to understand what is resistance, ऐसा क्यों होता है, and there could be many different reasons, and these are the few primary mechanism. It could be कि प्रोटीन जो है उसका फंक्शन उसने अपने आप को बदल लिया है। सो कि नेचर इज प्रोटीन जो दे आर वेरी क्रिएटिव, आपने जो दिया लगाया उसका थोड़ा मॉडिफाई, हल्का सा मॉडिफाई कर गया, अब टारगेट के ऊपर drug is not working. Could be one reason. It could be, for example, efflux. This is apne, I don't know, because you're not biologist, you probably know. Do you know what is PGP pump? Okay, so PG pump is a protein जिसके कहते हैं that pump efflux. जो pump है वो जो proteins हैं, they identify some foreign entity, they throw it out. तो drug आता है, वो drug को बाहर फेंक देते हैं. So this PGP pump efflux होता है बाहर पड़ गया तो ड्रग पहुंचा नहीं टारगेट तक. That could be another mechanism. There could be, there is passive diffusion. There could be there is a DNA damage हो गया है, it's not prepared. So there are 6 variation. ड्रग एक्टिवेशन अंदर जाने के बाद में ड्रग का स्लाइटली मॉडिफाई हो गया, मेटाबोलाइट बन गया अंदर के एंजाइम्स के कारण, इट्स नॉट वर्किंग. So there are 6 at least reasons which have been identified that you see I have mentioned here: epigenetic reflux, DNA damage, cell death, EMT, epithelial to mesenchymal translation, which is means the drug goes from the cell of site to away from there. It is EMT, MET coming back into. So drug target alter हो गया है, change हो गया, drug is becoming inactive. So there could be many, and there are encyclopedic information available on each one of these, but there could be many reasons for which it's not working. So in my lab we started to study looking at paclitaxel. I think I was telling yesterday about one third क्यों काम करते हैं, क्योंकि paclitaxel was in 1950s Research Triangle Park, North Carolina, there's this national lab, 2 chemists, Mansukh Wani from India, Gujarat, 1950s में, and Munro from New Zealand. They were both working there. I mean, US land of immigrants. वो lab में जाके वो दोनों काम कर रहे थे. Mansukh Wani was in Munro's lab. His whole job was to extract natural products from around the world, जहाँ से भी उनके हाथ लग सके, और उनको उस समय whatever few cell lines were available, just testing those cell lines. the compounds, extract, not the individual compound. Remember, 1950s, ये technique chromatography ये सब कुछ available भी नहीं थे. So they will test against the cell and see whether it's working or not. Anyway, long story short, they they tested many thousands of these extract from various plants from around the world. So my lab, for example, used to be at National Cancer Institute. National Cancer Institute has natural products. वहाँ पे जो chemists हैं, they work with people pretty much around the world. वो एकदम पाताल तक जा रहे हैं, ओशियंस के अंदर जा रहे हैं और वहाँ से नेचुरल प्रोडक्ट निकाल रहे हैं. वो कहाँ कहाँ अमेज़ॉन में जा रहे हैं, वो हिमालय में जा रहे हैं, ढूंढ ढूंढ के वो निकालते हैं, फिर उनको टेस्ट करते हैं. And there is one of the largest collection of natural products वहाँ पे. So these are what natural product chemists they do. So anyway, they looked at and they identified a compound which was from the bark of the yew tree, state of Oregon, Seattle के नीचे जहाँ से अभी मैं हूँ, स्टेट ऑफ ऑरेगन में. तो वहाँ से एक ट्री उनको मिला, उसकी जो बार्क है, उसके जो स्किन है, बार्क है, उसके एक्सट्रैक्ट से उन्होंने जो किया तो कंपाउंड दैट एक्सट्रैक्ट स्टार्टेड वर्किंग. तो इट शोड वेरी गुड रिजल्ट्स. दिस इज़ फ्रॉम 50 से लेके मैं आपको एक आध मिनट के अंदर पूरे 40 साल का 35 साल का काम बता रहा हूँ. तो दे एक्सट्रैक्टेड, दे कोलैबोरेटेड, they sent their compounds to National Cancer Institute in Washington, in Bethesda, Maryland area. Those compounds were tested. Or eventually it went through preclinical studies जो I've been telling you, preclinical studies develop in उस समय 80s में पूरे 70s में 80s में जितना पता था उसके बेसिस पे, क्योंकि आपकी मॉडर्न क्या है, मॉडर्न जो आज है कल ये पुराना हो जाएगा क्योंकि कुछ और चीज़ें आ जाएंगी. So at that time, whatever the best of knowledge was available, they used that, they employed that, and there were 50 clinical trials funded by the government were run. Eventually that compound came, and this is the compound, this is the structure of paclitaxel. Now if you look at the paclitaxel structure, there are so many chiral centers. It's not easy to synthesize a pure isoform. I mean, it can be synthesized. At that time there was no synthesis available also because we didn't know the structure kya tha. So then government floated, दो टेंडर निकालते हैं, BMS, Bristol Myers Squibb gave the best tender. They got to start making clinical trials were, और 1992 के अंदर the compound was approved, which became Taxol or paclitaxel जो है. This was for ovarian cancer for women. Compound gets approved 92 के अंदर और 94 के अंदर the reports come कि it's really not working. It works only in a small percentage of women. What happened? 40 साल लगाए, billions of dollars लगाए, जितना उस समय मॉडर्न था वो टेक्निक्स को, उस नॉलेज को यूज़ किया, but it's not working. It works only a small fraction of patient. Okay. Subsequently, so obviously the best of the brains, biologists, toxicology, pharmacologists, chemists, they got involved. कि हुआ क्या? Why it's not working? Now let's try to find out. It turned out, so one of the person who was very active and was the toxicologist on there was Susan Horowitz. She was a faculty in New York. Started look at ये है क्या? The drug was developed for targeting beta tubulin. DNA damage करता है, वो DNA को damage करता है. That was the presumably mechanism of the drug. And now these are beta tubulin जो हैं, these are जो आपके cell body में सेल के अंदर, you'll start to appreciate कि नेचर कितनी मज़ेदार है, कितनी ब्यूटीफुल है. सेल के अंदर एक प्रोटीन है ये जो 23 nanomolar, you can't even see through your eyes. There are 3 types of tubulins, alpha tubulin, beta tubulin and gamma tubulin. Gamma tubulin का फंक्शन कम है. Alpha, beta, gamma is based on their structure, orientation, कौन से एसिड, अमीनो एसिड कहाँ लगे हुए हैं. So and that combined forms what is known as microtubules. Those microtubules are formed. उन microtubules के सैकड़ों फंक्शन हैं सेल के अंदर, टिश्यूज़ के अंदर, ऑर्गन के अंदर, बॉडी के अंदर के, इसलिए हम सब अपना अपना काम कर रहे हैं. So that's only one of the proteins. So just imagine how many different types of proteins are in a cell and each one connecting with each other. It's an ocean. Cell is an ocean of protein. So it was developed for this beta tubulin. So and now there are, I'll come back to this. There are 28 drugs in the market trying to target beta tubulin. Anyway, and I'll come to that. So when she started, she and her team and other those biologists who were involved started to look at the drug was supposedly, so let me again show you this structure. ये beta tubulin, alpha and beta जो है, see I have written there alpha and beta tubulin form, they come together and form these tubules, columns बनाते हैं. They beta tubulin जो है, उसके अंदर जो कुछ एक proteins हैं, वो थोड़े बाहर को expose होते हैं. Now each tubulin, alpha tubulin, has many different types of protein just to make alpha tubulin. Each of those protein, so as a chemist to appreciation के रचनाकार की रचना कैसी है, एक बीटा ट्यूबुलिन जो है, जो कि स्मॉल एक प्रोटीन है विद इन द सेल, बाकी प्रोटीन्स की बात नहीं कर रहा, that is formed by 3 alpha beta gamma tubulin. Each one of those is a sequence of amino acids. उसके अंदर सीक्वेंस का भी किस ऑर्डर में है, that gives the shape of alpha beta gamma, right? Alpha and beta, they have kinase, GTPase, different type of proteins within that protein to form the bigger protein. And so they come together to form एक तरह की tube, nanotubes, microtubules उसे कहते हैं हम. And alpha and beta वो एक दूसरे से tightly packed होते हैं. उसमें से कुछ एक proteins हैं, naturally that's how it works. But some of this, and I'll come to that, let me not jump ahead of me. So beta tubulin को target करने के लिए presumably this drug was which works on beta tubulin. जो paclitaxel है, presumably taxanes work on यहाँ पे, you see my pointer here, वो यहाँ पे काम करते हैं. That was the understanding when it was developed, which is still correct. But the question is, जो single beta tubulin है, it turned out it has 6 different isoform. वो 6 different तरीके से orient हो सकता है, just single beta tubulin. Depending on which patient, उसमें कौन सा isoform of tubulin is overexpressed, this drug will work or not work. So beta tubulin 1, beta tubulin 2, beta tubulin 3, beta tubulin 4, 4A and 5. There are 6 isoforms. So and then she studied one of these studies to show किस तरह से कौन सा isoform ऊपर या नीचे होता है, किन which patients responds and which patient do not respond. They started to look at उन patient samples को, which ladies responds and which don't. It turned out the ones who did not respond to the drug had beta tubulin 3 and 4A, उनमें वो ज़्यादा overexpressed था protein, and those were about 2/3 of the women, those who were not responding. So we started to see what influences beta tubulin 3. So my lab, from looking from the chemistry angle, we started collaboration with somebody who was a tubulin expert, someone who had MD and PhD, clinician and scientist, his whole career was only in beta tubulin. We started to look at कि beta tubulin क्यों नहीं काम करता, और specifically beta tubulin 3 now, not just other beta tubulin 3 isoform. Just to give you an idea and appreciation कि how nature is complex, just beta tubulin, ये उसका जो मैं आपको दिखाने जा रहा हूँ is the simplified version. This is the simplified version of that. Now this single beta tubulin has many GT tubules which play in microtubule dynamics के अंदर उन proteins का role है. There are at least 19 different kinases जो उसमें involved है. ये सब मिला के एक beta tubulin 3 सिर्फ बनाते हैं, बाकी हजारों protein की तो बात में होगी. So how complex it is. So it's a challenging task. So we started to look at it, how do we understand? There are no tools available, how do we understand? So we started another journey, try to look at patients का sample लेके, and to realize to understand कि उनका makeup क्या है. No, this one cut. Yeah, don't show this one. Okay.